So what's happening specifically regarding the detection of pheromones? What are the systems that detect pheromones? How is this information processed within the brain? And how are behaviors, specific behaviors generated? How does an animal know that the signal come and lead to fighting behavior or mating behavior? How is the quality of the pheromonal information received, perceived, and how does it lead to behavioral changes? As I mentioned just before, there are these two signals, the two systems, the Vomeronasal system on one hand, the olfactory system on the other hand, and the assumption for a very long time was that the Vomeronasal system was specialized into the detection of pheromones, and the olfactory system was specialized in the detection of odorant chemicals. And this notion came from surgical ablation experiments in which people had surgically ablated the olfactory epithelium, and this led to impairment of odorant detection, or surgical ablation of the vomeronasal organ, and this led to defect in mating or aggressive behavior, and therefore presumably the detection of pheromones. So surgical experiments, surgical ablation were a big cue into the role of each of these two separate systems, and also the notion that these two systems share the work between cognitive smell and instinctive smell also originate from the study of the central projection of this system. So the olfactory epithelium is connected to the main olfactory bulb, and in turn to a number of nuclei in the brain that are together forming what is called the primary olfactory cortex, and then the information is distributed very widely within cortical and neocortical areas of the brain, and therefore leads to this cognitive perception of a smell. And in contrast, information that is detected by the vomeronasal organ seemed to be processed by an entirely different and independent central pathway from the vomeronasal organ to the accessory olfactory bulb to then specific areas of the medial amygdala in the limbic system that are themselves connected to specific areas of the hypothalamus that are specialized in aggressive and triggering aggressive and mating behavior. So specialized in reproduction and social behavior in general. So it seems therefore to make sense that this area that are involve the primary olfactory cortex and then higher cortical areas would indeed be responsible for the cognitive detection of smell, whereas areas of the brain that are part more of the limbic system, the amygdala and the hypothalamus, are more involved in processing pheromonal signal and the trigger of reproductive and aggressive behavior. So these seem all very logical, and at the molecular level, it was also very interesting to discover that neurons of the main olfactory epithelium signal through a set of channels that are cyclic nucleotide gated. Therefore, the signal transduction of olfactory signal use cyclic nucleotides that in turn lead to the opening of ion channel and therefore enable the translation of the binding of odorant to the receptor into an electrical signal, a change in membrane potential. And in contrast, in the vomeronasal organ, we don't find any functional cyclic nucleotide gated channel. What we found several years ago in collaboration with Emily Lyman and David Corey is the very strong and specific expression of a distinct ion channel called TRPC2 that is again very highly and specifically expressed in the vomeronasal organ and is responsible for the VNO signal transduction. So at the molecular level, we have these two ion channels that are each essential for olfactory transduction and vomeronasal transduction. And these therefore provide a terrific genetic tools to investigate or reinvestigate, if you wish, the function of each of these two sensory pathways in the brain. So by genetic manipulation of the gene encoding the TRPC2 channel, we perform a knockout of the TRPC2 channel and therefore led to an animal generate a line of genetically modified mouse in which the VNO does not function because the TRPC2 channel is non-functional, is mutated, and therefore the entire vomeronasal pathway is made non-functional. And therefore this mutated animal, this mutant, doesn't have a functional vomeronasal organ, is unable to detect pheromones, and we can therefore investigate the physiological role of the vomeronasal organ in the animal physiology and behavior. And similarly, by looking at the knockout of the cyclic nucleotide gated channel, we and others have been able to investigate the behavioral function of the main olfactory system. So this is just to show you the expression of this TRPC2 ion channel. What you see on this part of the slide is a section through this tubular structure that is formed by the vomeronasal organ. And what you can see here is the neuroepithelium that bordered lumen through which the pheromone arise in contact to neurons. And in red is an immunostaining with the TRPC2 channel, and you can see that the protein is highly expressed and very specifically expressed along the sensory terminal of the VNO neurons. Here seen even better on a dissociated neuronal preparation. You can see the sensory dendrite here where the sensory, the receptors and the channels are. So you know, these expression pattern really suggests an important role of the TRPC2 channel in the sensory transduction in the vomeronasal organ. And the idea is that the two families of putative pheromone receptor V1R and V2Rs, when binding to a pheromonal signal, lead to a signal transduction cascade and in turn to the opening of the TRPC2 channel. So by the knockout of the TRPC2 channel, one can completely abolish the signal transduction and lead to an animal without a functional vomeronasal organ. So this is what we did, and the first experiment that we performed when we obtained a mutant animal is to indeed validate the claim that the TRPC2 channel is essential for VNO signal transduction. And the experiment, so here's just the demonstration that in the TRPC2 mutant, there is no TRPC2 protein made anymore compared to ubiquitous proteins such as beta-tubulin. And in collaboration with Marcus Meister, an electrophysiologist in my department, and Tim Holly, who was a postdoc in Marcus' lab, we performed the electrical recording of the VNO neurons in response to pheromonal stimuli. So the idea is to use a flat electrode array in which each of the dots here represent a different electrode that can record from neurons the electrical activity of neurons in the vicinity. And a VNO epithelium is pressed flat against this electrode array and maintained by a mesh, and then we can puff pheromonal stimuli and record the activity of the neurons that have been stimulated by specific chemical cues. And when we did the experiment and compared the situation in the wild-type animal or the heterozygous animal to the situation in the mutant, it became very clear that pheromonal stimuli leads to an increase in the spiking rate of VNO neurons are recorded by the electrode array, but there was absolutely no stimulation in the TRPC2 mutant. So in other words, the VNO neurons is unable to respond to pheromonal stimuli. We know that there are neurons here here, because if we stimulate the preparation with potassium chloride, high concentration of potassium chloride, we can see a very strong, non-specific neuronal firing that comes just from the depolarization of the cells. However, these cells are unable to specifically respond to pheromonal stimuli. So the VNO is basically silent, and what we have is a mouse line in which the olfactory detection can occur, but the Vomeronasal detection is completely impaired. So what's happening to the behavior of these animals? Well, to our big disappointment at first, these animals didn't seem to show any phenotype. We expected from surgical experiment that animals without VNO would be unable to mate. But when we put a male mouse in the presence with a female, a male mutant in the presence of a female, they were mating perfectly normally, in fact, exactly with the same frequency as wild-type animals. So we were very disappointed and even questioned what the Vomeronasal organ good for. And then we thought a little bit further and decided to study another set of behavior. And we use very well-known observation from Conrad Lawrence that describe the behavior along these words. If you put together into the same container two sticklebacks, lizard, rodents, rats, monkeys, or boy who have not had any previous experience with each other, they will fight. You can add to these two politicians, two scientists, two whatever. When you put two males of any animal species in the same cage or room, they will tend to fight with each other. Well, we did this experiment, and together with the wild-type male mouse and mutant male mice. And I'm going to show you the behavior of the mutant compared to that of the wild-type. The behavioral paradigm that we use is as follows. We know that in rodents, fighting behavior between two males arise from the detection of male pheromones. So in order to set up an experimental system in which we can control the presence or not of the male pheromones that trigger the male behavior, our paradigm was chosen as follows. We had a resident male, so an animal that stayed in its cage for a couple of weeks and kind of established its territory. And we then introduced into that resident cage an intruder. And the intruder is of different kind. We first introduced a male intruder that is a castrated male. Pheromones are under the control, pheromone production is under the control of testosterone, and therefore the castrated male is unable to produce any male pheromones. And when we do this experiment, you can see in this video, so you have the resident male, and the castrated animal further here. The intruder does not emit any male pheromones, and as you can see, these two mice just coexist very peacefully. So there's not even really seem to be any specific behavior of one animal versus the other. Now, in the next video, what you're going to see are the same two animals, but the experimentator has put now 10 microliter of male pheromones on the fur of the castrated intruder. So this castrated intruder does not naturally emit any pheromones, but the male pheromones is added exogenously. And when this is done, you have now the intruder here. As you can see, the resident detects the male pheromones and immediately start to fight. So the resident has adopted this defensive posture, doesn't understand what's happening to him. And as you can see, the resident is really very aggressive, and this is an extremely robust behavioral reaction, which is a male detecting another animal emitting male pheromones will very brutally attack that animal. Okay, so in the next So that was the positive control. This is what wild-type mice do. A male mouse detecting another male using olfactory cues will attack that other male. Now, if we use a TRPC2 male, and so this is the male animal, and this is the same intruder here that has been swabbed with urine, what you see is strikingly different. And I hope that even people without any experience in male behavior, in mouse behavior, can very well visualize that what we have here is absolutely not a fighting behavior, but instead a very surprise mating attempt of the male mutant versus the other male. So this was extremely puzzling, extremely surprising. The male mutant, instead of attacking the other male, is trying to mate with it. So what's going on? Well, the key experiment was to actually put both male and female in the same cage. So again, if you have a male mutant in the presence of a female, the mutant will mate perfectly normally. But if you put also a male in the cage, a male intruder, what we discover to our big surprise is that the mutant is unable to discriminate between males and females, and in fact attempt to mate with each of them with equal frequency. And that led us to propose that the role of the vomeronasal organ is not to trigger mating behavior, as was what was expected from the literature. An animal without a functional VNO seemed clearly able to mate normally with a female, but these animals seem completely unable to discriminate between males and females. And so we even control for this behavior in a large arena that you can see here. So this is result from the observation that social behavior in general can be very different in small cages or in more natural conditions. So we put a bunch of male mutant in the cage and left them for several weeks, just letting them do whatever they wanted for extended period of time and recording them constantly. And as you can see when we play the video, is this male that formed these courtship chains that are quite striking, in which one male is trying to copulate with the male in front and is trying to be copulated with the male just behind. Those are extremely striking behavior that can go on for several minutes. Now, I'm showing this for entertainment value, but also for a very interesting purpose also, which is that these courtship chains that are observed in the mouse are actually strikingly similar to the courtship chains that have been observed in drosophila in a particular mutant called the fruitless mutant. So fruitless is a transcription factor that has many splicing variants. Some of them are sexually dimorphic, and the mutation of the male-specific splicing variants lead to these male flies that show these male-male courtship chains that are indeed very similar to what we've observed in the TRPC2 mutant mice. Now, this is very striking because fruitless is a transcription factor that is expressed very widely within the brain, and is thought to be responsible for the development of the neuronal circuit that enable courtship behavior. And the TRPC2 channel is an ion channel expressed only in sensory neurons that give information about the gender of the animals. And I really very clearly find the similarity of the behavior very striking. Obviously, it's a very interesting question of whether or not the mammalian brain is expressing a fruitless equivalent, and so far nobody has really been able to find interesting candidates. So from this study, we propose a model of the control of mating behavior, reproductive behavior in a mouse that is quite different from the classical view of the role of the vomeronasal system. What we found is that sensory cues that are independent from the vomeronasal system are sufficient to trigger mating behavior. And the role of the vomeronasal organ is to provide another type of information, which is gender identification. So here, there are really two systems that work with each other. One is the vomeronasal information that provide information about gender, and the other one, some other sensory signal that is sufficient to trigger mating behavior. And very clearly, in the absence of vomeronasal information, the default behavior is mating behavior. And when a male is encountering another male, then the vomeronasal cues detect signals that say no mating behavior, but instead aggressive behavior. And so this obviously is quite different from the classical view of the role of the vomeronasal organ in triggering mating behavior. And I will come back later during the talk on some possible explanation of the discrepancy between the results obtained with the TRPC2 channel, so genetic ablation, compared to what has been obtained with classical surgical ablation. So I'll tell you a little bit why I think those results were different. And this comes from a result that we obtained very recently and that I will describe in the third part of the talk. Now, this is obviously very striking, this shared work between vomeronasal system and other sensory cues in the control of gender identification. And at that point, I really was curious on how are other animals distinguishing the sex of their conspecific? So I went into the literature and investigated a little bit what people had described in other species. And here's what I found. So in this particular species of bird, the shell parakeet, this is a female, and this is a male. And the animal, the parakeets, recognize the gender of their conspecific based on the presence of these blue dots on the top of the beak. And this particular blue dot is essential for sex identification, which is that if you paint a blue dot on the beak of a female, the animal now is identified as a male, and other males will attack that female with a blue dot, thinking it is a male. And similarly, if you mask the blue dot from the beak of a male, then the other males are going to try to mate with this male without a blue dot by thinking it's a female. So the blue dot, the visual cues, the identification of this blue dot is essential to the identification of this animal as a male or female. Similarly, in this other species of bird, the American flicker, he is a female, he is a male, and what provides the identification of one being male, the other one a female, is the presence of the black mustache. So if you were to mask the black mustache on the male, this animal will be identified as a female, and the other males will try to attempt to copulate with this male without a mustache. And this animal here that is a female, if you paint a black mustache, males will attack that female thinking it's a male. And Tinbergen, who is a very famous ethologist, described this by calling those signs the badges of masculinity, which is here, this is my blue dot, or this is my mustache, I'm a male, and if I don't, then I'm a female. And so this is quite interesting, is a visual recognition of the gender identity. And I think that what we found for the pheromones is the olfactory equivalent of the badge of masculinity, which is the vomeronasal organ is responsible for discriminating between males and females. Now, obviously, the animal we care the most about are humans. What kind of strategy do humans use? And is the vomeronasal organ also being used for sex identification? And here things are likely to function very differently. The TRPC2 channel, which is responsible for the function of the vomeronasal organ in rodents, is a non-functional gene in humans and actually in higher primates. These little triangle that you see here, there are 9 of them, are the site of deleterious mutation, such that higher primates and humans have a number of deletion or frame shift or nonsense mutation that make this gene unable to generate a functional protein. And it's quite interesting to actually look throughout evolution when this mutation occurred, and this is the work of the laboratory of Emily Lyman, that shows that the mutation really start to accumulate at the split between New World monkeys and Old World monkeys and apes. So all this part of the tree of higher primates really is unlikely to use the vomeronasal organ as a tool to discriminate sex. And I think what's quite interesting and was proposed by Emily Lyman's group is that these split here between the New World and the Old World monkeys also correspond to the duplication of the red and the green opsin genes, such that animals in this part of the tree here have an additional opsin receptor gene, and therefore the ability now to discriminate between two colors, red and green, whereas animals in this part of the tree have one opsin gene that detects both green and red color, and so this is being perceived as one particular color, whereas here those animals can discriminate between these two wavelengths of photons that are very close to each other, but if distinguished by distinct receptors, can now appear as distinct colors. And these, from an evolutionary point of view, can provide an enormous advantage. For example, the ability to discriminate between a ripe and a non-ripe fruit. Ripe fruit is full of calories, full of sweet, and this is obviously very advantageous for animals that are able to distinguish those highly nutritious food from a non-ripe fruit that doesn't have all these properties. So to come back to our two systems, we've seen using genetic mutation that the vomeronasal system is specialized into detecting the sex identity of individual animals, and that the dichotomy between pheromone perception in the olfactory, in the vomeronasal system, is not that absolute. In other words, that mating behavior can occur without the vomeronasal organ. Therefore, there has to be something else that provide information to the animal about the presence of conspecific and the ability to mate. Now, how are we going to go explore the system further? Well, as I mentioned, we and others have identified the specific receptors for chemical detected in the vomeronasal organ, and Richard Axel and Linda Buck discovered the olfactory receptors responsible for detection in the main olfactory system. And so one really interesting goal is to try to understand what are the chemicals that are detected and provide animal with information about the gender identity of the animal, or which one provide information that leads to aggressive behavior or any type of behavior that is triggered by these two systems. And these direct question, what ligand generate what type of behavior, is a little bit difficult to address right now because of technical difficulty into expressing pheromone receptors in vitro, and therefore finding an easy high throughput assay to identify the ligand of these receptors. And so what the strategy that my lab decided to use is start in the center of the brain instead of the peripheral organ. So instead of trying to identify what are the receptors involved in specific behaviors and then trying to go and follow the circuitry in the brain, we decided to do exactly the opposite, which is, it is well known that specific nuclei in the hypothalamus are involved in aggressive behavior or reproductive behavior, and we therefore decided to trace the input to those specific centers. In other words, what are the areas of the brain and what are the specific neurons of the vomeronasal organ olfactory epithelium that send input processed by different brain circuitry that end up in, let's say, an area of the brain involved in reproductive behavior or in aggressive behavior. This requires two parameters. One is the type of neurons that in the brain that we want to investigate. So a specific set of neurons that are clearly involved in either reproduction or the control of aggressive behavior. And the second parameter is to find a tool that enables to link this particular set of neurons to the connected circuit in the brain. So the set of neurons that we decide to study first are neurons that express and release a very particular neuropeptide called luteinizing hormone releasing hormone. This is a neuropeptide that is expressed by a very small population of neurons in the medial preoptic area in the hypothalamus. They may be as few as 600 or 700 of these neurons that are dispersed within this very large area of the hypothalamus called the medial preoptic area. These neurons synthesize LHRH and release it in the portal vein, and the neuropeptide LHRH is absolutely essential for the control of fertility and reproduction in vertebrates. So animals that are deficient in LHRH are sterile and do not develop their gonads, functional gonads, and are impaired in sexual behavior. The function of these cells is as follows. They release LHRH into the portal vein that will then interact with neurons, with cells in the pituitary gland, and lead to the release of LH and FSH that is in turn is released into the blood and lead to the development of the function of the gonads, both males and female gonads. These functional gonads will in turn release steroid hormones. These steroid hormones will, on one hand, lead to the development of the secondary sexual traits and also provide a feedback to the brain in both enhancing sexual behavior, but also providing a feedback to the release of LHRH. LHRH also directly communicate with other areas of the brain by synaptic contact and are essential for sexual receptivity and modulate sexual behavior. Now, what is quite interesting is that these neurons are really the master regulators of reproduction and fertility in the animals, and therefore their own function is very tightly regulated. They are sensitive to both the internal and external state of the animal. So one interesting factor that controls their function is actually a very unknown set of factors that is a developmental clock that triggers puberty. So these neurons are not functional before puberty, and then at some point they become functional and release LHRH at high frequency. The developmental clock that triggers this function is not well understood at all. But for sure, this is one of the main control of the function of LHRH neurons. Now, these neurons are also sensitive to external cues, for example, pheromonal cues or sensory stimuli that then leads to reproductive behavior. So it is known that pheromone detection leads to increased LHRH synthesis and increased LHRH release. Now, moreover, these neurons, the function of these neurons is also extremely sensitive to the internal state of the animal. For example, an animal that is starving is not going to reproduce. So an animal that is very stressed won't reproduce either. And this is because the other areas of the brain that deal with stress or the level of nutrition are sending signals that tightly control these LHRH neurons. So, you know, the hypothalamus in general is in charge of the homeostasis of the animal and the appropriate coordination of all the functions of the organism, reproduction, aggression, nutrition, sleep, etc. And so it's very essential that the release of LHRH has many levels of control, both by the environment and the internal state of the animal. So we decided that these neurons would be a perfect target of our study, and that by investigating all the nature of the sensory cues that send input to those, we will understand better the circuitry controlling reproduction and fertility in the rodent brain. Now, the second set of tools that we used are viruses, and in particular a set of viruses called Pseudorables viruses that have the ability to replicate within neurons and, more importantly, to jump across synaptically connected chains of neurons. So it will replicate into a neuron, then cross the synapse and reach from postsynaptic to presynaptic cells, and then jump again, etc., and will therefore infect all the neurons that are connected to each other synaptically. Now, the type of virus that we use are conditional Pseudorables viruses that have been made by Lynn Inquist in Princeton, and also Jeff Friedman at Rockefeller. They built a modified Pseudorables virus that, when infecting the neuron, is non-functional. So that virus, shown here in red, infect the neuron, but is unable to replicate. And when a neuron expresses a particular enzyme called a Cre recombinase that is indicated here like a pair of scissors, it cut out a cassette out of the virus genome that make now the virus able to replicate and also to express the green fluorescent protein, and therefore now the virus can replicate, jump across synapses, and label all the infected neurons in green, fluorescent green. And therefore we generated a transgenic mouse line expressing the Cre recombinase specifically in neurons expressing LHRH. So LHRH promoter driving the Cre recombinase. And by injecting this conditional virus in the medial preoptic area, the virus will infect neurons but replicate and become green fluorescent only in neurons expressing the neuropeptide LHRH, and therefore one will be able to visualize very nicely all the LHRH neurons, but also all the afferent neurons, the neurons synaptically connected to these LHRH neurons. And so when we've performed the study, we identify a lot of infected neurons throughout the brains, indicating that they were synaptically connected, sending information into LHRH neurons. And as control, we could visualize a number of brain areas that were known to send information to LHRH neurons. In particular, areas that provide sensory inputs, that provide information about the circadian clock in the suprachiasmatic nucleus, that provide information about stress level in the brainstem, or the level of nutrition in from the arcuate nucleus and other areas. So we found all of these by injecting the virus by LHRH neurons. We were able to identify all these brain areas, but our main goal was to try to investigate within the Vomeronasal system all the specific brain areas that ultimately send input to the medial preoptic area, the neurons expressing LHRH neurons there. And specifically what we were hoping is that if the virus could indeed jump from postsynaptic to presynaptic cells throughout jumping enough synapses, we could even be able to recognize specific population of the Vomeronasal organ and maybe recognize them with the green fluorescent protein, maybe identify what are the specific receptors that ultimately send information to LHRH neurons. And when we performed the experiment, when I say we, actually my graduate student, Haiyan Yoon, the result was quite astonishing, which is that we recognize many brain areas, but within overall the olfactory and vomeronasal system, the brain areas that we recognize in the cortical amygdala, the piriform cortex, the olfactory tubercle, the olfactory bulb, and a specific population, also very small population of neurons in the olfactory neurons. Now, what's wrong here? Well, what's wrong is that all of these belong to the main olfactory system, and we were actually completely unable to recognize, to identify any label area within the vomeronasal system, which is that in contrast to what was found in the literature, in which classical dye tracing experiment had identified strong connection of the medial preoptic area to the medial amygdala and the vomeronasal system, what we found was instead very strong connection to the main olfactory system, and in particular to cortical areas of the olfactory system. So what's going on? Well, you know, classical dye tracing experiment put a dye in a particular area where the LHRH neurons are located, them among many other types of neurons. And these tracing experiments indeed show very strong link between the vomeronasal system and the medial preoptic area in which LHRH neurons are located. Our experiment is a genetically controlled tracing experiment in which what we visualize are the very specific connection of this very precise population of neurons expressing the LHRH genes. And when we do this, what we found is that it's possible that all the other neurons around are connected to the vomeronasal system, but the LHRH neurons precisely are not connected to the vomeronasal system and instead are connected to the main olfactory system. So this is quite interesting, and in particular, it points to a particular population of neurons in the olfactory system that send input to LHRH neurons, to neurons involved in the control of reproduction, and therefore these neurons are very likely to detect pheromones. In fact, by definition, if they are connected to areas of the brain involved in the control of reproduction, they are pheromone detecting neurons. We've recognized that these neurons expressing LHRH, which is essential for the control of reproduction, have this massive connection from the olfactory system. This is obviously an anatomical finding, and it's absolutely essential to have some type of functional correlates. In other words, if indeed the main olfactory system provides such a massive input to LHRH neurons in the medial preoptic area, then animals that are deficient in olfactory function should also have deficiency in reproduction. And this is indeed exactly what we found. Remember, we found that the TRPC2 mutant male are perfectly able to mate, but mate both with males and females. And when we now investigated male mouse that are deficient for the olfactory cyclic nucleotide gated channel, so impaired in the main sense of smell, what we found was that these animals are absolutely unable to mate. So they don't even recognize the female, don't even acknowledge the presence of females. So these provide a very nice and direct functional correlate to this massive input from the main olfactory system into the control of reproduction. So in other words, it's now very clear that both vomeronasal system and olfactory system both contribute to the perception of pheromones that lead then to the control of the reproduction and fertility in the animal.